Hello everyone, welcome to BrainMap. Sorry. This seminar series is co-sponsored by the P41 Funded Center for Mass-Scale Mapping housed in the Martinos Center. It is our great pleasure to have Dr. Shan Siddiqi today to give us BrainMap talk. Shan Siddiqi is an assistant professor of psychiatry at Harvard Medical School, neuropsychiatrist at Brigham and Women's Hospital, and the director of psychiatric neuromodulation research for the Center for Brain Circuit Therapeutics. His research is focused on causal mapping of human brain function and dysfunction. Using techniques such as functional connectivity MRI, his lab maps brain circuits to link brain lesions and brain stimulation sites that can modify different psychiatric symptoms. These circuits can be then targeted by the treatments such as transcranial magnetic stimulation, TMS, and deep brain stimulation, DBS, to alleviate symptoms in psychiatric disorders. His work has been recognized by multiple awards from Brain and Brain Behavior Research Foundation, the American Neuropsychiatric Association, the American College of Neuropsychopharmacology, Harvard Medical School, and many others. Again, thank you very much for giving us talk today, and the stage is yours.
Great. Thank you for the invitation. I appreciate it. I think the talk is unmuted. So again, thank you guys. My name is Shan Siddiqi. I'm a neuropsychiatrist at the Brigham, and I'm here to primarily talk about deriving brain stimulation targets using causal mapping of human brain function. I'll talk a little bit about what I mean specifically about causal versus the way that we do things traditionally. Here's my disclosures, where the grants come from, and some commercial and clinical consulting. By far, my biggest funding source is the NIMH, so I'm definitely biased in favor of circuit-based dimensional models of mental illness. Now, I have a causality joke, but I don't really know if it's the reason why you're going to laugh in a few seconds. See, that was a high-risk joke because I wasn't sure if people were going to get it, and I didn't know if people would laugh. But the funny thing is that you're now laughing at the fact that I'm making fun of the fact that I made a prediction about your laughter earlier. And I think the reason I illustrate that is if I had said at the beginning that I'm going to predict that you will laugh at my joke, that implies that I'm going to predict that you're going to laugh because my joke is funny. That's not the reason why you're laughing, right? You're laughing because my joke was actually really, really lame. And it was kind of funny because you're laughing at me. So this is why prediction does not equate to causality. And I think this is an important distinction I'm going to talk about a little bit more here. But here's another example of that. Another huge study found that cell phones cause cancer. You know, what was the WHO thinking? And if you look at this this correlation plot here, it looks a lot like a resting state time series. He emphasized, well, you're not. There's so many problems with that. He said, no, just to be safe, until I see more data, I'm going to assume that cancer causes cell phones. You can draw either conclusion from this data, and this is the where we have to exercise caution when drawing causal conclusions from correlated data. But the broad reason why I'm talking about this topic is to think about how we conceptualize brain circuit therapeutics. Like, how do we use brain imaging to find treatment targets? And I'm going to focus on depression here, because that's probably the disorder that's been studied the most using this approach, but a lot of this applies to other disorders also. So, for example, this dates back at least to 1937, when James Papes described a brain circuit for emotion, which we now call the limbic system. Some people say it might date back 1,000 years before that, when the Middle Eastern physician Ibn Sina described that manipulating different parts of the brain or different parts of the skull can cause different brain functions. Or even 1,000 years before that, when the Greek physician Galen pointed out that different lesions can cause different symptoms. But I'll start in 1937. In the 1950s and 60s, a couple of neurosurgeons, including Jeffrey Knight in London and Thomas Valentine here at Mass General, started disrupting this circuit using different neurosurgical techniques like subcaudate tracheotomy and anterior cingulotomy. And it worked. Their basic idea was that if patients are too emotional, let me break their emotion circuitry to see if they get better. And they seem to get better. In the 1980s, Robert Robinson noticed that lesions to the left dorsolateral prefrontal cortex might cause depression. So lesions to the limbic system are treating depression, but lesions to the prefrontal cortex are causing depression. And subsequent functional neuroimaging studies showed the same thing. Depression was associated with decreased activity in the left DLPFC, an increase in the limbic system. So starting in the 90s, Mark George, Eric Wasserman, Alvaro Pasqualeoni, and some others at NIH started treating depression using transcranial magnetic stimulation, or TMS, to the left DLPFC. The idea is if breaking the spot gives you depression, if depression is associated with decreased activity in this spot, then let's stimulate it and see if we can get depression better. This ended up becoming a pretty effective treatment, and it's now an FDA-cleared treatment for depression, which is used all the time. There's a very busy TMS clinic here at the Martinos. And about 10 years later, Helen Mayberg found that deep brain stimulation to the subgenual cingulate cortex, part of the limbic system, can also treat severe refractory depression. This finding, they're still figuring out the optimal way to replicate this, but the same basic idea. If the limbic system is overactive, then let's try suppressing it. DBS is believed to be inhibitory. Potentially the most recent exciting development in this field was in September 2022, when the FDA cleared this novel rapid-acting TMS protocol for depression. This is 10 treatments a day. Each treatment was triple the dose we were previously giving. So we're giving 30 times the dose that we were previously giving every day. And we do that for 5 days. And it turns out that we can get people better. We're seeing the best remission and response rates that we've ever seen in psychiatry in a week. There were two key optimizations here. One was the dosing pattern. Like I said, they gave 30-fold the dose times 5 days. So a total dose of 5-fold what was previously being given in a month, but they're only giving it in a week. And they were seeing better remission rates than they'd ever seen before in just 5 days. But the second optimization was this includes personalized image-guided targeting for major depression. And I highlighted this is for major depression because I think this is the tip of the iceberg. This tells us that we can modulate depressive symptoms with TMS very effectively and efficiently now. For a long time, TMS research was slow because the effect sizes were small. So if we got patients a little bit better and it took a month and a half, we didn't really know if we got them better or if they got better for some other reason. But when you can get them drastically better in a week, all of a sudden you can test whatever your circuit hypothesis is, and you know the answer pretty quickly. So if we can find the right target, we can test this for a lot of different things, but it becomes a question of finding the right target. Have any of you guys seen the TMS clinic here or been there, seen how they work? Maybe not. This is how we do it. We use scalp landmarks to identify a patient's TMS target. So you use a pulse over the motor cortex to make their thumb twitch. Then you pull out a tape measure and you measure 5 or 6 centimeters in front of that, and you say that looks like about the right spot. So in clinical practice here in the most medically advanced city in the world, we're using a tape measure to target electromagnetic brain stimulation. Same technology this guy's using to measure this dog. That's how we target it. We could be able to do it. We could do better. We could be more precise, but we do this for a reason. These landmarks are based on locations of lesions that cause depression, but also because this treatment works. And what people said was, well, you know what, we're going to pursue practicality over perfection. Let's just get it out there. Now, but the problem is that ever since then, the same landmarks have been used for PTSD, generalized anxiety, addiction, et cetera, with mixed results. And it makes sense. Why are we using lesions that cause depression to target treatment for addiction? We should be able to do better than that. We have the ability to target very precise locations in a patient's brain with TMS using the stereotactic neuronavigation system. But all the precision in the world is useless if we don't know the right spots to target. And so we don't use this in our clinic. We use it in clinical trials. So we need a reliable framework to start translating lesions to TMS and DBS targets for different disorders. And in our lab, we do this using data science mostly. We're starting to translate this into clinical trials now. Now, what do I mean when I say targeting? This is what it kind of looks like. And this is the rig. The blue thing on the person's head is a TMS device. It applies stimulation with a radius of about 1 centimeter. And if you give it repetitively, it has long-lasting effects. Those little gray balls are trackers that a 3D camera can sense, so it can figure out where the coil is and where the patient's head is. It registers all that into a piece of neuronavigation software, comparing it to their MRI scan. And we can get within a couple of millimeters of precision as to what our intended target is. And that's useful because, like I said, the TMS coil, the field is about 1 centimeter. So a couple of millimeters of precision is good enough. But how do we find that target? Well, traditionally, brain lesions have been used to study causes of neuropsychiatric symptoms. And the way we've done that is we localize a bunch of different lesions. Let's say these 3 people had strokes. We look to see where those strokes intersect. And we say, if all these people develop the same symptom, then that must be where that symptom came from. And let's call it depression. That's where depression lives in the brain. And that's how brain mapping was done until the 1970s. This is a Google Ngram search of all the different brain mapping studies until over time. And until the 1970s, it was mostly brain lesions and brain stimulation intraoperatively. That's how they mapped the brain. Then functional neuroimaging came around, and this happened. That all of a sudden, functional neuroimaging became the predominant way that we were doing brain research, or structural neuroimaging, or the types of correlative neuroimaging. And this is great because it allowed us to do experimental manipulations that we couldn't possibly do. But at the same time, what's interesting is that the lesion and stimulation studies went away. People started replacing it rather than augmenting it. And so we started replacing our causal research with correlative research because we had abilities to test different kinds of hypotheses. Eventually, brain stimulation studies made a comeback, but brain lesions never did, even though it was a valuable source of information. So there is some use of these studies still, and I'll show you how. So I'll show you this image again. This is the ideal situation when we are trying to do a lesion study, but often in the real world, it looks more like this. You have some lesions that intersect, some that don't, and they cause the same symptom. And so when you look at the overlap, you say, well, maybe the lesion location matters, maybe it doesn't. Maybe stroke just causes depression no matter where you have a stroke. And that's where the field was sitting for a long time until we started bringing in the human connectome and using it as a wiring diagram. They say, well, maybe all these all these strokes that cause the same symptom have a similar connectivity profile, either with structural connectivity or with functional connectivity. They might overlap with the same brain network. And if that's the case, as this audience will know, we've got this wiring diagram of the brain that's largely mapped here. If that's the case, if breaking this circuit causes depression or any other symptom, then in theory, stimulating the same circuit should relieve the same symptom, in theory. But the question is, does that actually work? Is that actually going to help us find treatment targets? So let me see if I can close this little message box here. Oops. I guess I can't close it. So so we started looking at this in a study a couple of years ago where we tried to map a human depression circuit based on focal brain lesions. Took all the lesions we could find, looked at their connectivity, and compared the connectivity of lesions associated with depression to the connectivity of lesions not associated with depression. And this is what it looked like. The peak was right around here, which just so happens to overlap with the most common TMS targets for depression. So we said, oh, great. Now we have some sort of idea. TMS is working by modulating the same network that, when damaged, causes depression. And this was interesting, but around the same time, in the same issue, Amit Etkin wrote this editorial where he pointed out that it remains to be shown that lesion or stimulation anywhere within a particular network has the same downstream effects on behavior or symptoms. Like, it remains to be shown. He called us out. What he pointed out was, and he was right, was that even though we showed that stimulating that circuit seems to relieve the symptom, we're not really demonstrating causality unless we can show that stimulating a different circuit does not relieve the symptom. We have to be able to show specificity, which we didn't do here. And so this raises the question of how do we define causality in general, because that study wasn't perfectly causal. In general, there's only one way to define causality with certainty. There are a lot of ways to estimate it, but the only way to define it definitively is to give an exposure, measure an outcome with that exposure, get in a time machine and go back in time, and then don't give the exposure, then measure the outcome without the exposure. And of course, we all know this experiment's been done before, but it requires 1.21 gigawatts of power. And so we know that either you have to harness lightning, which is dangerous, and they don't allow us to have a research assistant to do that kind of stuff anymore, or you have to get Libyan terrorists to steal plutonium for you. And I was born in Pakistan. If I work with terrorists, they're not going to let me on a plane anymore. So instead, what we do is we estimate causality in humans using natural experiments whenever we can, followed by randomized trials. In medical school, they teach you the gold standard for causality is randomized trials. But before you can do a randomized trial, you have to have a causal hypothesis. And for example, the first ever randomized clinical trial was streptomycin for tuberculosis. When they planned that trial, they already knew that streptomycin kills tuberculosis in vitro studies. They already knew that mycobacterium causes tuberculosis based on Koch's postulates of microbiology. And then they did the clinical trial. In psychiatry, we've been doing it differently. We've been throwing treatments at different disorders. For example, we say, this works for depression. Let's see if it'll also work for addiction, without a clear causal hypothesis. And so that's That's what we need to do a little bit differently, go into the causal hypothesis first, and we can do that with some natural experiments. By the way, I have to clarify, this isn't the same Mike Fox that I usually cite. It's a different one that you might remember. But there's the rest of the Fox citations are going to be to a different Mike Fox. Now, how do we actually estimate causality? We wrote a review article on this last year that you can check out in the corner there. But here's the basic premise. One is to see if there's a counterfactual. If you stimulate one thing versus not stimulating it, does that cause a difference? A second thing to look at is specificity. If you stimulate one circuit versus stimulating a different circuit, do you get different effects? Another thing is if you experimentally manipulate active versus sham, do you get different effects? Dose response is a really strong one. When you give more of it, does it have a different effect? Coherence is a particularly strong one. When you see when you stimulate something versus lesioning it, you get opposite effects. And if so, that really strengthens your causal inference. And reversibility is another important one. When you stimulate something and then stop the stimulation, does that make the effect go away? There are at least 10 criteria for causality depending on who you talk to. So at least 9. The Bradford Hill criteria, the most commonly used. But we adapted these to brain imaging because many of those criteria were sort of unified across modalities. But these are the specific criteria that differ according to different brain imaging modalities. And here's how they differ. There's just a brief summary of how different modalities are increasingly causal. Of course, most of us know that task-free neuroimaging is not causal. If you add tasks, you can test some causal hypotheses, but not very well. Atrophy gives you a little bit more information, but it doesn't give you clear temporality because when atrophy happens, you don't really know if the atrophy caused the symptom or there was some other thing that caused both the atrophy and the symptom, or maybe if the symptom caused the atrophy. Lesions, on the other hand, are acute. So you can see no symptom, then lesion, then symptom. So that gives us better temporality, which is a mandatory criterion for causality, according to the Bradford Hill criteria. And then we can get progressively better in this framework, and the best we can do is convergent causal mapping. In other words, if you take multiple causal approaches like lesions, stimulation, atrophy, even correlative neuroimaging, and they all converge on the same answer, then each method will mitigate the confounders from the other method, and then you can really draw strong causal conclusions. Again, we published this entire framework last year, so you're welcome to check it out if you're interested. But here's how we started by testing that. We localized a bunch of incidental brain lesions and see if how they relate to symptom outcomes. Do the same thing with TMS sites. I told you earlier that we're very imprecise with our tape measure approach to targeting TMS. As a result of that, we get incidental variability in where patients are actually targeted. So then we can use stereotactic neuronavigation with an MRI to localize the incidental stimulation site and compare that to the outcome, and we have a natural experiment with pseudo randomization. But it's not pure randomization. There's still some confounders here. But deep brain stimulation, we get the same issue. On the top right there is 27 deep brain stimulation electrodes in the subgenual cingulate that were implanted by the same neurosurgeon. And they were all in 27 different locations because there's incidental variability. So these 3 different sources of incidental variability, if they converge on a common circuit, then that gives us a lot of information about the causality of that circuit, because even though each of these approaches has some confounders, they have different confounders. So if they all give you the same answer, then we've addressed all the confounders. And so that's how we propose addressing causality in general. And here's how we did that. We localized a bunch of incidental brain lesions, some all the way from Vietnam veterans and penetrating head trauma to a bunch of ischemic strokes, all the way from St. Louis to Melbourne, 50 hemorrhagic strokes from Chicago, about 150 different TMS sites, all the way from Boston to Melbourne, 44 deep brain stimulation sites in patients with major depression from Atlanta to Berlin, 32 sites in patients who received deep brain stimulation for Parkinson's, which is a commonly used treatment, but in this case, they also had depression measured before and after. And same thing with thalamic DBS sites for epilepsy, also a commonly used treatment, but in this case, they also had depression measured. So we took all this information to see if we can get it to converge. And here's how we do that. We localize the incidental brain lesion. We trace it onto a standard MNI atlas brain. We use a normative connectome database to see what it's supposed to be connected to. Now, this is an important step here. We're using a normative connectome database, not individualized connectivity, because you can't take individualized connectivity from a lesion because the lesion's gone. So here what we're doing is we're assuming that people's brains are more similar than they are different. That assumption is not perfect. We know there's individual variability, but we're not accounting for it yet because the variability in the lesion location is so large that it dwarfs individual variability in connectivity when we've tested that assumption before. So we start by deriving the normative circuit, and then I'll show you later how we individualize it. Then we see if the patient developed depression or not, and we put that together into a map of the connectivity of lesions that cause depression. We do that for 5 different lesion data sets. Same thing with TMS. We localize a bunch of incidental TMS sites, see if the patient, we'll see what it's connected to, see if the patient's depression got better or not. We get a map of the connectivity of TMS sites that relieve depression for 4 different TMS data sets. Same thing with deep brain stimulation, localize the stimulation site, see what it's connected to, see if the patient's depression got better or not, and we get a map of the connectivity of DBS sites that relieve depression for 5 different DBS data sets. So we had 14 data sets with 14 heterogeneous sources of information. So at this point, I was talking to Mike Fox, who was my primary mentor on this project at the time, and he said, why don't you dump them all into the same analysis and see if they converge? And I said, okay, you don't understand. I'm a psychiatrist, he's a neurologist. In psychiatry, things don't replicate or converge, right? We're not used to that. Like you're used to neurology where things make sense. So that's not going to work. But it turns out he was right. This is what we saw. On the left here is the connectivity of lesions that cause depression. In the middle is the connectivity of TMS sites that relieve depression. They're relatively inverted because lesions are believed to be inhibitory and TMS sites are believed to be excitatory. So that's why we inverted the colors to make it line up. The connectivity of DBS sites that modify depression. And these are more similar than expected by chance. We test that using spatial correlations with permutation testing. I'm happy to explain the math if anybody's interested. You can't see the p-values here. But do you know if it's possible to get rid of this little thing here? I'm trying to just close it here and it doesn't work.
Stop screen sharing.
Oh, okay.
And then,
Oh, I see.
screen sharing.
Okay.
No,
Yeah.
Yeah.
Okay. Well, maybe this thing up on top will disappear in a second. You'll be able to see the p-values. But the point is these are more similar than expected by chance. And we do that by looking at the spatial correlation of each map with every other map. And then you can't just do p-values on a spatial correlation because there's a spatial autocorrelation, which will lead to a false positive. So instead, what you do is you recompute the spatial correlation after randomly shuffling each patient's clinical outcome for the different patients' neuroimaging. And then you get a distribution of what's expected by chance. And so you look, well, what percentage of the time is the real correlation stronger than the permuted correlation? And that's how we calculate these p-values. I love actually being able to talk about that. Usually I talk to psychiatrists and I just say, I'll skip the math for now. But by looking at that, we show that these things are more similar than expected by chance. As expected, then, they also predict clinical outcomes. So this is the map that I showed you earlier, except all 14 data sets are put together. But if instead of making this from 14 data sets, we make the map from 13 data sets and use it to predict outcomes in the 14th, leave one data set out cross-validation, this is what we get. Stimulation site connectivity to the leave one data set out circuit significantly predicts outcomes better with, and that works with real stimulation, but not with permuted simulations. Again, you can't permute the imaging itself because that doesn't solve the spatial autocorrelation problem. But when you permute each patient's imaging with a different patient's clinical outcome, it does solve that problem. So we weren't sure if we believe this still yet because this was a retrospective analysis. So we then replicated it in an independent set of patients with multiple sclerosis. On the left is the same type of network that was derived from 283 patients with multiple sclerosis. The patients who developed depression were more likely to have, sorry, it's okay. Thank you. So if patients with more severe depression were more likely to be connected to the circuit, and we can do this, we can actually predict how severe their depression is going to be based on how much their lesion overlaps the network. What's interesting is these people have white matter lesions, not gray matter lesions. So there's an active debate of whether bullet signal in white matter is useful or not. Yes. Well, by definition, multiple sclerosis is white matter lesions. So gray matter. Know this. So the atrophy it happens in gray matter, right? But the lesions tend to happen in the white matter. Okay, well, I'll defer. You clearly know more about it than I do, so I'll defer to you. But what the reviewers complained the most. The biggest complaint from the reviewers was that how do you believe anything in white matter in the white matter? Many of the lesions were the white matter, but I didn't look at all of them individually to see which ones were where. Yeah. That's interesting. I didn't realize that. So with the imaging, with the 3T imaging that we have, can we detect the gray matter lesions at all? Okay, so we had 3T imaging in all these people, so it looks like the white matter lesions might be correlated with what's happening in the gray matter. So maybe I shouldn't draw that conclusion that it's about white matter then. Thanks for the comment. But sorry. Yeah. I guess maybe if we had 7T imaging, we could see if it's driven by gray matter or white matter. So I'll refrain from making that conclusion then. But the point is that we replicated the thing in an independent lesion data set. And it still lines up with TMS and DBS. But no, thank you for that comment. We also found in the MS data set that it's specific to depression. So we had 12 different metrics on the neuro quality of life scale. And it turns out that connectivity of the lesion to our depression circuit predicts depression better than it predicts 10 other control metrics. And it negatively predicts positive affect, as you would expect. The peak in the MS-driven depression circuit was here in the ventral midbrain. We're pretty close, overlapping mostly with the ventral tegmental area. Green is the peak spot. Blue is a mask of the ventral tegmental area. So it kind of makes sense that it's a source of dopamine in the brain. And so interestingly, nobody's ever tried, there's never been a trial of dopaminergic antidepressants in MS. So this is one of the things that we propose here. So let me summarize the first few things I just told you. TMS, DBS, and brain lesions can all modulate a common brain circuit. And lesions can help us find new TMS or DBS targets potentially for multiple disorders. But I'm saying potentially for multiple disorders, but I only showed you data on depression. So we've extended that further with Parkinson's disease also. We originally submitted this to Nature Medicine, and they rejected it and came back and said, how can you claim that you developed a transdiagnostic tool when you only tested it on depression? So we use Parkinson's disease because the next most common use of brain stimulation, DBS is commonly used for Parkinson's. And here's what we get. Here's the connectivity of lesions that cause motor symptoms of Parkinson's, of DBS sites that relieve motor symptoms of Parkinson's. And this is the location of the most common clinical trials. There are 10 clinical trials of TMS for Parkinson's that were positive, and they stimulated that spot in the motor cortex. We don't have individual variability in stimulation size, so we couldn't derive the full map. This analysis is not as strong as the depression one, but it suggests that this doesn't just work for depression, it works for other things too. Those are just the first couple of things that we looked at, but since then, it's been shown in a lot of other ways too. Lesions causing amnesia are connected to the same network as DBS sites that cause cognitive side effects in Parkinson's. Lesions causing tics are connected to the same network as DBS sites that relieve tics in Tourette syndrome. Lesions causing seizures are connected to the same network as DBS sites that reduce seizure frequency in epilepsy. Same is true for vertigo with intracranial electrical stimulation sites, tremor with DBS sites, and addiction with TMS sites. So at this point, we've got these 6 examples as well as the 2 I showed you earlier. So at least 8 examples in which lesions help us find better stimulation sites when we bring the connectome in as a filter. So I'm going to zoom in a little bit to this addiction example. On the left here is the normative connectivity of stroke lesions that lead to remission of nicotine use. And on the right here is the connectivity of penetrating head trauma lesions that reduce the future risk of alcoholism. And again, these are more similar than Shan. So I'm going to say that again. If you have a stroke that makes you less likely to smoke, it tends to be connected to the same circuit as a penetrating head trauma lesion that makes you less likely to drink. And again, these are more similar than Shan's. So the question is, how is that therapeutically relevant? Well, on the left here is that cravings circuit. That's what I like to call it, since it's sort of common between nicotine and alcohol. We're not sure if it's involved in cravings, but that's a hypothesis. In the middle here is the electric field distribution of the H4 coil, which is a TMS device that is FDA cleared for nicotine use disorder. They didn't prospectively target the circuit, but it just so happens that their target overlaps. And on the right is the electric field distribution of the H7 coil, which is a TMS coil that was recently shown in a multi-center clinical trial to be effective for alcohol use disorder. So it seems that targeting this circuit lines up with the results of clinical trials, even though those clinical trials weren't designed to target this circuit. So let me summarize the last few things I just told you. Lesion connectivity can be used to validate existing brain stimulation targets, from depression to addiction. But that was validating existing targets. Can we actually use it to find a novel brain stimulation target rather than just reinforcing what we already know. So specifically, we looked at that in anxiety. Anxiety disorders are the most common class of mental illness and the world's sixth leading cause of disability in humans. I believe it's even more common in cats, but I'm not sure the data are out yet. Despite that, they've been shown to be underdiagnosed and undertreated. Personally, I think the reason why we don't do a good job of treating anxiety disorders as doctors is because we have anxiety disorders ourselves, and we see our patients with anxiety disorders and we're like, that seems totally normal. But in fact, it's one of the world's leading causes of disability and is known to be undertreated. There have been no new FDA-cleared treatments for anxiety in the last 20 years. The only FDA-cleared treatments we have for anxiety are repurposed antidepressants and sedatives. We need to be able to do better. We've got new treatments for depression, but nobody's really working on anxiety. And one of the reasons why it's difficult to study is because it's highly comorbid with depression. So every time we study anxiety, we have to control for depression to look at the independent effect. And brain stimulation seems to help anxiety broadly, but of course it does because it helps depression when they're comorbid. So the question is, how can we disentangle these two things from each other? And so we did the same stuff that I was showing you earlier, except with anxiety. And from now on, when I say anxiety, I mean anxiety independent of depression. So every time we look at anxiety, we're controlling for comorbid depression. On top is the localization of a bunch of incidental TMS sites and a bunch of incidental lesions that were associated with anxiety. This was again, localizing them on a standard MRI template brain, looking at their normative connectivity, see if the patient developed anxiety or not. And across each of these 4 data sets, we get a map of the connectivity of lesions or stimulation sites that cause or relieve anxiety. The one on the left, we already published in AJP in 2020, but everything else that I'm about to show you is unpublished. This was for each data set, but when we put it together across all the data sets, all these 4 data sets, we had 2 data sets of TMS sites and 2 data sets of lesions. On the left is the connectivity of TMS sites that relieve anxiety. On the right is the connectivity of lesions that cause anxiety, independent of depression. Again, we would expect these to be inverted because TMS is believed to be excitatory and lesions are believed to be inhibitory. So that's why the left is relieving anxiety and the right is causing anxiety. Again, these are more similar than expected by chance, using the same stats that I was describing to you earlier. Happy to answer more questions about that if you guys are interested. So one question, aside from showing that, okay, lesions can actually be used to identify a better TMS target, can that actually be personalized? That's another question that's been active in the field right now. So the short answer is yes, but I'll show you how we figured that out. This is a clinical data from a clinical trial in which 300 patients were all stimulated at the same spot with structural neuronavigation. And they all got baseline resting state fMRI scans. They weren't good quality fMRI scans. They were 5mm spatial resolution and 10-minute acquisition. But there were 300 patients, so it was a large enough sample to overcome some of the noise. So we can compute the individualized connectivity of each TMS site and see if the patient developed anxiety or developed improvement in anxiety. And again, put all that information together into the map of the connectivity of the individualized connectivity of TMS sites that selectively improve anxiety independent of depression. Again, this is significantly similar to the normative TMS and lesion-derived anxiety circuit that I showed you a minute ago, suggesting that if you individualize this network, you can further improve our efficacy using resting state fMRI. Just out of curiosity, I looked at biomarkers. This is exploratory. This wasn't a priori. But it turns out that resting state connectivity within our anxiety circuit significantly predicts treatment-induced change in anxiety, more so than any of these Yao control networks. And you might look at this and say, well, this looks like the default mode network. It must be the same thing. But connectivity within the anxiety circuit significantly outperforms connectivity within the default mode network as a predictor of anxiety outcomes. So if we take all these 862 patients that I just showed you and put them into a single convergent map, we get a peak in the superior frontal gyrus and the lateral parietal lobe. Again, it looks like the default mode network for the most part. And this is what it looks like. So if you have a stimulation site connected to that circuit, you're more likely to have improvement in anxiety. If you have a lesion connected to that circuit, you're more likely to have worsening in anxiety. But still wanted to validate it a little bit more with DBS, because I've been telling you earlier, the more different modalities you can use to validate something, the more you can assess causality. Because you control for more and more confounders that way. So here's what we did. We localized a bunch of incidental DBS sites in patients with Parkinson's. They get stimulated at the subthalamic nucleus. We had 74 patients. Assessed their overlap with the anxiety circuit. And this illustrates why I like subthalamic DBS as a model, because it straddles the task positive and task negative networks. You can see that some of the DBS sites are overlapping with the yellow and some are overlapping with the blue. That's just an incidental thing that happens with subthalamic DBS. So let's just look at these sorts of models. So then we look to see how much a patient's DBS site overlapped with the anxiety map. And it turns out that significantly predicts whether the patient develops anxiety or not from the treatment. This is a known side effect of DBS, and it's something that DBS doctors try to avoid. They don't know why DBS causes anxiety, but it seems that overlap with this circuit is what actually predicts whether the patient develops anxiety or not with DBS. This is also specific to trait anxiety relative to state anxiety. So let me summarize the last few things I just said. Lesion-derived circuits can give us better targets for brain stimulation. Personalizing those targets can further optimize efficacy. Then there might also be a few other approaches to personalizing. So I'm going to spend about 5 minutes telling you about other things that we're working on. One is based on personality traits. So this is my student, Stefan Palm. This is not published yet, but it's in bioRxiv. We looked at lesions that modify novelty seeking. So patients who had a glioma resected and had novelty seeking as a personality trait measured before and after the glioma resection. And we had two independent data sets. The connectivity of lesions that modify novelty seeking in data set 1 are on top, and then data set 2 are on the bottom. And again, these are more similar than expected by chance. Novelty seeking is an important personality trait because it's seen in a lot of things, in impulse control disorders and personality disorders. So if we can find a TMS target to mitigate that, that'll be valuable. Interestingly, recently another preprint came out showing that stimulating a similar circuit modifies a personality trait that they call hypnotizability. I think that overlaps with novelty seeking. And the point was not about a specific personality trait, but the fact that you can actually modify somebody's personality with a targeted brain stimulation. Now, whether that's a good thing or not, we don't know, but of course we first have to figure out how to do it, then we can decide if we should do it or not. There are other dimensions we can use also. This is my psychiatry resident, Andrew Pines, is working on lesion network localization of psychosis or detachment from reality. And perhaps not surprisingly, lesions that cause psychosis are consistently connected to the hippocampus. Even if you drop all the hippocampal lesions, they're still consistently connected to the hippocampus. There's another student of mine, Bea Milano, who's working on perceived disability independent of objectively measured disability. And the region that seems to be popping out is a region that's been implicated in alien limb syndrome previously, which is a syndrome where you feel like you've lost control of your limbs and somebody else is controlling your limbs. The same lesions connected to the same network seem to make people feel that they're disabled, even when they don't have any objective measurable disability. There's another student, a grad student, Summer Franson, is working on clustering out positive versus negative affect. And lesions that modify positive affect seem to be connected to the VTA, to the ventral tegmental area. Again, not a surprise, the source of dopamine. So this is all great, but this is all normative models. We talk about stimulating the dorsolateral prefrontal cortex to modify depression and other things, but that's not one thing. This audience will know that this target is quite heterogeneous within individuals, but heterogeneous between individuals, but it can be consistent within individuals. For example, on the top here is an individualized resting state network map of my brain, and the bottom is a colleague of mine. We were both fourth-year psychiatry residents at the time. And you can see in the dorsolateral prefrontal cortex, not knowing anything about what the colors mean, the topography is different. These are different networks. So for example, red here is the dorsal attention network. If I were to stimulate the dorsal attention network in my brain, I would hit the purple stuff in his brain, which was language. And so this further justifies why we might want to personalize things. So one way to personalize is based on functional connectivity. We've thought for a long time the connectivity of the subgenual cingulate is what helps us find the best TMS targets. So we identified personalized TMS targets based on subgenual connectivity here, negative correlation. Compared them to the patient's actual TMS site, measured the distance between those two things. And it turns out the closer that a patient's TMS site is to the optimal target, the better they do in terms of depression. And this is not the only time this has been shown. There are like 6 studies now showing the same thing. And this was independent of optimizing the normative site. This is the independent effect of individualized connectivity. So this is one thing that individualized connectivity can do, just simple seed-based connectivity measurements. Of course, we can also map resting state networks in individuals. Again, I think a lot of the people here probably are doing resting state connectivity research, but for those who aren't, we can individualize brain mapping algorithms have become increasingly common these days. We call it precision functional mapping. For example, you can estimate connectivity between any 2 points in the brain, or between hundreds or millions of different points in the brain. And there are certain brain regions that reliably cluster together, even at the individual level. And if you look at that and plot it back onto the brain, these regions that cluster together kind of look like you would expect. There's a default mode network, there's a motor network, there's a visual network, et cetera. You can train a machine learning algorithm to then recognize and label which network is which. And so that's what we use in this study I'm about to show you now. So on the top is a group mean network map from the Yao Atlas, which again, I'm sure you guys are probably familiar with since it was developed here. On the bottom is an individualized brain network map of the same networks. Again, there are a lot of different kinds of precision network mapping techniques that can be used to individualize network maps. I don't prefer any one over any other one, but there are a few differences that I want to highlight here. On the top is the DLPFC, our usual TMS target. At the group level, there's nothing there in the dorsal attention network. At the individual level, there's a big spot there. And if you guys look at a lot of these, you'll know what I'm about to tell you, which is that almost everybody has a spot in the dorsal attention network somewhere in the DLPFC, but it seems to be in different spots. So when you average it out, it washes out, but it's always there. Same thing with the default mode network in the subgenual cingulate. I'm drawing your attention to the subgenual cingulate now. That's a region that's been implicated in depression. Some people it overlaps with default mode networks, and some people it doesn't when you do these individualized maps. So what does this mean? This might tell us something about biomarkers. So you guys might have seen Hisheng Liu and Justin Baker's study recently, or I think Danhang Wang was the first author, showing that this helps us predict symptom phenotypes in schizophrenia and bipolar disorder. But it can also teach us more about diseases that we didn't know about. So we looked at this with traumatic brain injury and depression after traumatic brain injury. Clinically, as neuropsychiatrists, we believed for a long time that this is a distinct syndrome, but we'd never proven it. So we looked at the connectivity between these regions of interest that I just showed you after individualizing the network boundaries. And this only works if you individualize the network boundaries. If you don't individualize the network boundaries, the thing that I'm about to tell you doesn't work. Here's what we found. Connectivity in the circuit was the same in patients with versus without traumatic brain injury if you're not depressed, but it went in opposite directions if you are depressed. In other words, TBI-associated depression was physiologically different from primary major depression. It wasn't just the sum of the two, they went in opposite directions. And then we replicated this in an independent cohort, so a total of 273 patients. So we believe this is real. We're starting to call this TBI affective syndrome, that this is a different thing, not just TBI-associated depression. I don't like the term TBI-associated depression because it implies it's an incidental comorbidity. I think it's a distinct syndrome. This also predicts therapeutic response. So we found an independent clinical trial of TMS for TBI-associated cognitive symptoms. And it turns out TMS site connectivity to that same dorsal attention network predicted mood changes. Everybody was stimulated at the same site, but the more they were connected to the dorsal attention network, the more they had improvement in mood, even though half of them weren't even depressed in the first place. And that was true for with active stimulation, but not with sham. So it seems like this correlative metric that I just showed you, when we add a causal metric, we can push it along the causality continuum I was showing earlier. Now, does it actually work? We also did a pilot clinical trial of this approach for TBI-associated depression. It was a small sample, and we did fancy targeting versus sham. We didn't do fancy targeting versus simple targeting. And the reason we did that is because no other intervention has ever been shown to work for these patients. So the first thing we wanted to do is see if we can just get them better. And if we can get them better, then we'll figure out why we got them better. And in short, this is what happened. On average, patients went from marked to severe depression all the way down into the mild to moderate range, or even remission range with real stimulation and not with sham. Again, it's a small sample. We didn't assess the effect of targeting, but around the same time, a different TMS trial came out, did an almost identical treatment protocol, except they didn't do individualized targeting, and that trial was negative. So it might be that the targeting matters. So let me summarize the last few things I just told you. TBI-associated depression is a distinct physiological entity, not just TBI plus depression. An abnormal circuitry in this TBI affective syndrome can be localized and targeted. So let me show some conclusions. I'm going to summarize everything I just told you. The TMS and DBS can target the same circuit to treat the same symptom. Lesions can be used to identify TMS and DBS targets. Depression severity maps to a similar circuit in major depression, penetrating TBI, stroke, epilepsy, Parkinson's disease, and now also multiple sclerosis, which might be gray matter. The results were derived from causal sources of information. Different depressive symptoms, such as anxiety versus dysphoria, seem to map to different circuits. And other disorders also seem to map to specific circuits, like addiction, TBI, affective syndrome, as we're calling it. We've also done this with other things like PTSD, psychosis, etc. And personalized brain mapping might help us further improve the efficacy of these treatments. So, of course, a lot of people helped make all this happen. I'm going to stop here and ask if you guys have any questions. And of course, this is how you can reach me if you want to try to find me later. Thank you.